Yes, we're now having our fourth interview together with Lorenz Adlung. Lorenz Adlung is a visiting scientist working at the Weizmann Institute for Science in the field of systems biology, and therefore he's combining computer science and biomedical research to understand complex phenomena of life and health and disease. And he has earned his doctoral degree at the German Cancer Research Center and has been a member at the iGEM Team Heidelberg 2013, as you can also see on what he's wearing today. I think he's going to be able to tell us a bit about the modeling they have been doing in the iGEM Team 2013. And besides all of his research, he's also a science communication enthusiast and has participated in over 100 science slams. And I think we're excited to hear about that as well.
Definitely. So usually science and results are in the foreground, and science communication rather plays a second and not so important role. This is why we decided today to turn it around and start talking about science communication. So this leads me to our very first question. What do you think is the best way to reach as many people as possible with science communication?
First of all, pleasure is all mine to be here. So thanks for setting this up. I'm not exactly sure your question is the right question, to be honest, because I'm not sure it's about, reaching as many people as possible. Because you might speak to a crowd of 100 people, and you make quite an effort, to convince them that, science is important and your research matters and stuff like this. But if you reach one of them and convince this person that science matters and your research is important, then I think it's worth it. Okay. And then, of course, you can say the more people you reach, the more likely it is that, you always have like 1% that you can convince, and therefore then you convince more people. But that's not always true, because at some point, the crowd gets anonymous because it's just too big. You cannot really identify individual people anymore. You cannot, on a personal way, interact with them. So I think it's a trade-off. I think, in general, we need to try to reach as many people as possible, also as diversely as possible, because if you only use like one channel, then maybe you always get people who go to this kind of event, like a science slam, or people who use Instagram or whatever. But only if you spread it, you also can address a certain diversity that represents and that is present in our society. So to answer your question, how reach as many people as possible, just do different formats. I mean, because there are so many ways, right? There are so many ways you can express yourself. There are so many ways you can explain your science. It can be something online. Okay, nowadays, I always call it the guerrilla tactics of, just going on the street and catching people waiting on a bus stop, involve them in a conversation, in a nice way. It's not really feasible these days, but, there's many different ways. You can do it at university, with new students, try to get them involved a bit. There's different levels. You can talk in your family, once they get a feel of, you're always the guy who comes to a party and then, talks science, then this is also a way, to connect. So if you want to reach many people, be diverse, but, just be prepared. This is some lesson that I had to learn the hard way early on. You won't convince them all, and they won't be all your friends after this event. I mean, usually people now, Science Slam, it's very popular and people look positively on it. But there's always people who are skeptical. There's always people, against the mainstream. You see it now with all the education that we have to do when it comes to viral pandemics. There's always people who they will never believe you. They will never be convinced, no matter how many data you accumulate, no matter how many facts speak in favor of your point of view. You cannot convince them. They will always exist, okay? This is a given. But if there's this one person that you convinced, that was not believing in science or not trusting science or, was having prejudice, one person, then this is a big achievement because, one can make a difference, and then one, also spreads the word. And it should be worth it, just doing it for a single person, I think. So don't get depressed if, first you don't get the resonance, you don't get all these likes and comments that you would like to have. There's even people who still tell you that you're shit, you don't know stuff, even though you have a PhD in this topic and they hardly have like, finished high school, but whatever, they will tell you, you don't know nothing. Don't let it get, too close to you. This will always happen. It has nothing to do with you. And actually it's also, even the positive feedback is not particularly about me as a person or you as a person. It's about the science, and this is what it should be all about. It should all be about the science, I think.
Yeah, that's a good advice. Thank
you.
And talking about diversity, how do you differentiate between different age groups? Like talking to elderly versus talking to high school students? Maybe?
Yeah. I think age, experience is a function of age in a sense. So the older you are, the more experiences you got. So you always need to, this is anyways an advice I always give. Ask, if you are asked to give a talk, first question you should ask the organizers, who is the audience? And then you have to ask yourself, okay, what do they know? What do they not know? How can I catch them? And so if you talk to a younger audience, you may think, okay, they don't know like even simple arithmetics, maybe, in primary school and then, early high school. So I want to speak about mathematical modeling, ordinary differential equations. They don't know how to derive such an equation, how to integrate. So you have to start on a more conceptual level. But on the other hand, younger audiences, they are naive in the most positive sense. They don't know all these borders. They don't know these different disciplines and that they come with a lot of expectations. So they just ask these like very general questions that are even for us, who know a lot, are very challenging to answer. So take this into consideration that it's very easy to get a very curious audience excited, and that's my hypothesis. Maybe I'm wrong, but I think younger crowds, it's super easy to get them excited. And then, of course, if the audience is more mature in a sense, they have an idea of how science works, and there's this one field that is now super fancy, I don't know, single cell or whatever you name it. Then of course they're a bit more reserved, also because then they say, but this is more cool than this other research. But then you can also use this, and challenge them a bit and say, now I talk about, who does still talk about, I don't know, Western blotting, like bulk measurements of thousands of cells, protein is not cool at the moment. Everyone does, genetics and transcriptomics. But then you tell them no, for reason A, B, C, and this is then how you get them. So I agree, you need to address them separately, but it's not only a function of age. It's like where are they coming from? What are their questions? What are their daily lives? Because in the end, you always connect through experience and emotions. This is how people connect. And of course, emotions, they change with life. So younger people are different emotionally than older ones. Always, you should include also maybe a pun, or a joke in your speech. And then, of course, you also know, okay, maybe with elder people, I should not talk about the aging process in a too depressing way, maybe. But and when you're younger, school sucks, but science is still great. So how can I, maybe school doesn't suck because it provides you with all these general ideas of how to think to be a good scientist, and then suddenly it gets exciting again. So there's different ways. I just, I think the most important time for preparing a talk is actually before you start to even put something on a paper. Just first ask your question, who is the audience, and how to address them properly. The way you speak, even, of course, the way you speak is different if you talk to toddlers or to elder people, right? So, and at some point, you will do this, subconsciously, and we'll start with this. But at the beginning, I think you really need to reinforce this kind of thinking, okay, who is the audience? What is the message I want to deliver? And what's the right way to deliver my message to this audience, I think.
Okay. And do you think everybody can do science communication? Like, no matter which degree level one have? Like, do you have any advice for bachelor or master students who haven't conducted much of their own research yet?
Yeah, so this is a good question. Can everyone do science communication? I would say yes, because there is not this one format. There might be people, they don't feel comfortable in free speech, They don't feel comfortable being on stage. But maybe, this also just is something that you learn, and in the beginning you don't like it, but suddenly you get hooked and you get excited about this. But maybe you don't, and maybe you don't want it, and that's okay. All I'm saying is then there's maybe another format of science communication that you better like. You do animations, I don't know, you do sketches and drawings and more like science arts, or, you like to do YouTube videos, or God knows what, there's different ways. There's not just being on stage and doing Science Slam. There's not just writing a blog. Maybe you're into writing, so you write a blog, or I don't know, maybe you do these TikTok videos. So in a sense, I think it's pretty likely that there's at least one format of science communication that everyone would feel comfortable about it. And I think this is the only precondition of doing it. If you feel comfortable talking science, then there's nothing that can hold you back. And even, bachelor students, master students, I talk to them and they say, yeah, but we don't even yet have our own research. But maybe that doesn't matter. What matters is, do they have questions, right? Do they have a subject that they study? What are the questions in the subject? What are the methodologies they will soon apply? Maybe they don't know yet how to do it, but on a conceptual level, they can still talk about the subject, still talk about these questions. And, studying involves thinking. It doesn't only involve, experiments or computer simulations. So this is something they can tell the people outside, because they don't know. Maybe they didn't even study. Maybe they studied, other stuff like humanities or economics or God knows what. So there is something they have to tell, and if they feel comfortable about it, they should do it. They should do science communication. I'm not forcing anyone. I don't want, forcing them is certainly not the right way, because you will see it. You will see if people felt forced to do this sketch. You will see if people felt forced to write a blog. You will see it, of course, if they're on stage and they don't really like it. I'm just saying it can be super rewarding, and eventually now even, funding agencies, they start to acknowledge doing good ways of science communication. So I'm always giving this example that now I'm on a Marie Curie fellowship from the European Union, and in this application that you need to file, there's one paragraph that particularly asks, like, how do you disseminate your scientific findings to the lay public? And then if you can mention, I did a couple of science slams in the last years, this is, this is something unique. This is something maybe other people don't have. And then they say, okay, this is maybe really a good way to propagate. I mean, it's not propaganda, right? But to propagate your science and your enthusiasm for it. Because again, this is what matters in the end. This is how you connect to people. And I think it's a need, because, the money is coming from the people. So also the research should go back to the people in a sense.
Definitely. Well, that's a good advice. So you already mentioned it, the different formats. A Science Slam is a really good format, and as an iGEM team, we plan to host a Science Slam with other iGEM teams all over Europe, presenting their research. But obviously, Corona has forced us to modify our plans a little bit. So do you have advice for inexperienced speakers in general, concerning Science Slams in general, and specifically for participating in a digital form? Like, is something different?
Yes. So first of all, I think I also participated, even twice at the Science Slam that the iGEM team Bonn organized at the Caesar Institute. So I'm pretty sure they have an elaborate list of good slammers. And other than that, it's I can provide you with a few names that immediately come to my mind. But I think what's even better is if you tell people, to just see for themselves what they like, because it's very subjective in a sense. So there's like these organizations in Germany that sort of in a more country-wide manner, or like organize these science slams. It's scienceslam.com and scienceslam.de. You can find them, and they have, first of all, you see all these YouTube videos where you can get a feel, or also a simple way, because they are mostly in German. What's the idea of these online formats? Should it be in German or in English?
In English.
English, yeah.
And
So-
inviting other iGEM teams, which means not really professional ones, but people telling about their own research at iGEM.
Ah, fantastic. Yeah. So, then what I would recommend, because I can only speak for myself. So if you go to my website, lorenzadlung.com, and then there's this tab communication, and then they're all English, like all these different, there are some slams, there are some, free speech and stuff like this. And then you can go to, I did one for the trilateral one in Arizona last summer. It was organized between Russia, Germany, and the US. And if you click on my slam there, what YouTube does is it recommends you like similar stuff. And then, you can check stuff that is similar. You get a feeling of, topics and people and names. And I think towards the last part of your question, then getting people in a digital format is super easy because there's no barrier. You don't need, they don't need time to travel. So I would be surprised if this would make it harder to recruit good speakers. And again, there's many out there. I mean, now, Science Slam is really popular in Germany. So I think you shouldn't have a problem to find excellent speakers.
Okay, and yeah, of course, we also had a look at lots of Science Slams where you participated in. And when you think about all the events you had, can you think about a program part which worked especially well? And another question concerning that is, we found it really interesting that you use rap in lots of your Science Slams. Why is that?
Yes. So towards the first question, I think what's important indeed is the host of the event, like the moderator. Because I think it's essential to do a certain warm-up with the crowd. If they are sort of prepared and ready to be entertained, then it's much easier also for the speakers. Now, of course, in a digital format, it's a bit more problematic because they're not sitting all together. You don't see them. You don't know how they react. Actually, it's a big problem. I just had a few days back, I had a job interview, and they used the online tool where you could neither see nor hear the participants. So there were like 80 people watching me, and I was making jokes and trying to slowly explain difficult parts, and I had no idea whether my message was delivered, And I think this is really a challenge. So maybe I'm not sure what would be a proper way to ensure that you make the audience ready for getting these talks. So now some of these popular Science Slam formats, they also change to YouTube Live or Instagram Live or something, and they also still have a host who does in the beginning a bit of introduction, and then also does a bit of stand-up comedy. So maybe that's enough, but I'm not sure how they can really ensure that people are watching, people stay like on their screen, or maybe if you start with this sort of comedy, instead they will just procrastinate and open another screen and do something else, and you lose them there. I just don't know. But my feeling was really, if you have a good host who also then moderates questions and interacts with the speaker and with the audience, then this is really invaluable. So this is very, very important. And then, of course, with these events, you need to acknowledge sponsors and people who help with the organization. Usually, I think it's easier if you sort of distribute this throughout the event. So don't make a huge block in the beginning or the end where you say, okay, now I want to give you some, more bureaucratic advice, or, so try to, scatter it a bit, I think. Because again, if people are just on their screen at home, there's a lot of competition. If something gets too long, they will just, browse somewhere else. So I think that's a challenge. So try to make small, digestible blocks of good entertainment. Then this will work. And why do I do always this rap kind of thing? Well, I was listening to rap music even before I mean, I wasn't born a scientist, I guess, but, or maybe I was, I don't know, but very early on, I listened to rap music, and I really liked this sort of, I could really emotionally connect, you know, some rap music has really strong like emotions that are transferred, and I got hooked in a sense, and I sort of wanted to use this element to get people hooked to my research, sort of. And also, you know, the first science slam I did was, you know, on the blood that is flowing and flow cytometry. So there was a lot of flow, and in rap you also have like the flow of words, how you do the rhythm and speak and rap, whatever. So it sort of fit, and I really enjoyed it because this is something, you know, the audience doesn't expect. I mean, they don't know what to expect, but then usually, you know, it's always more or less like a funny, scientific, entertaining talk. But then suddenly, you know, the beat comes in and you start to rap, and they just look at you and you know, look at each other, and like you can really see that they are, you know, flabbergasted and shocked, or I don't know. And this is this very short moment, you know, it's just the blink of an eye, but this is really priceless. I enjoy this like every time. You know, and then it's sort of it became my signature move to always do this kind of rap thing, and yeah, I think it, you know, I'm not sure. I think there's even, you know, people who really don't like it and who don't get along with this, but I mean, this is my 10 minutes on stage, so I can do whatever I want, and this is the freedom that I dare to use as I like. So, and actually, just a couple of days ago, I learned that I will be funded, generously funded by the Bosch Foundation, to produce a science rap EP. So I will soon start producing an EP with, I don't know, maybe 5 or 6 rap tracks about scientific topics, about Corona as well, but also about my research, obviously. I might even feature some, you know, German rappers. At least that's what I wrote in the proposal. They say, you know, we still have to negotiate how to spend the money, but maybe there's even a budget for buying a feature. So this is pretty exciting, and I'm curious how this will work, because also there in the proposal, I had to think, how do you deliver this? Because, you know, usually you do some release party, and you invite people and do some sort of performance there, which now we can't know that it works. So do we also do some live streaming, or do we just, you know, just put it on, you know, online music portals? We don't know yet, but let's see.
That sounds really cool. So
yeah,
I think it's a good way to transport that message.
Yes. And okay, again, also when we speak about reward, right? This is basically the proof that you can get even, you know, funding for this to disseminate your research. And maybe I haven't stressed this enough. It's incredible if you are on stage, you speak about your science, you know, what is really close to your heart, what you really love, what you do all day, and then people applaud and they are cheering at you, and, you know, they are coming afterwards and say, wow, this was super interesting. I didn't know about systems biology before. Now I got hooked. It's super cool. You go to conference, you know, you give excellent talks there. It's still your science, right? So there's many parallels. There's an audience, you know, they are listening carefully, but in the end, you know, you will get this sort of modest applause at best, you know, and people ask this question where they are basically like, okay, so you did this, I do something else, have you also looked at this? And then you say, no, I mean, most likely not, but they are, you know, so this kind of appreciation is something you won't get in the scientific world, which is also okay, because, you know, science is a more serious business, and that's all fine, but appreciation can really, really help yourself, you know, help your self-esteem, help you, you know, through rough phases that you always have in the lab, you know, when things don't work. But suddenly you go on stage, you deliver your message that science is still awesome and people connect to this. And this is, again, priceless. Money can't buy this. And still, sometimes you get money for it, you know, research funding. So I think it's like a win-win situation either way.
Definitely. So earlier, you kind of mentioned it, when people are kind of hooked up with science, what are the things that science must offer to let them participate in science?
Oh, it must be open and freely available. You know, it's horrible. I just I started a podcast with a German writer. I mean, she's she has many talents, but first of all, she's a writer. And every episode, we sort of tell each other a story about biosciences. And usually this story is inspired by a publication. And then it's horrible because I tell the audience the story, but they cannot access the original publication because it's hidden behind paywalls. And this is a disaster because if I'm successful, you know, and they get hooked with science, but they cannot look at the original like data, they will again, you know, get suspicious for good reasons because they cannot see it, and seeing is believing after all. And if you know, withdraw this information or hide it for whatever reason, might it just be profits, you know, then it's like maybe you hide it for another reason, you know, maybe something is flawed there. And if people can't see for themselves, how can we convince them in the first place? And this is a huge problem. And I understand, you know, this is business models and big publishing firms, they rely on this. I'm just saying it's a huge problem for the credibility of the science itself and us as scientists. So, of course, what I always do when I publish stuff, I try to put all the data out there. I do actually. I put all the code out there, all like tutorials. I leave contact information so people can reach out to me, which they do. And it's not always easy because, you know, it takes time to curate data sets, to curate code so that people can follow it and reproduce everything. And even if you provide it, you know, I thought, you know, for my last cell paper, you know, I wrote this tutorial to reproduce all the figures and provided all the data, the raw data, the process data, you know, the final matrices. So you just need to run the plotting function. And still people send you texts and they don't understand. Sometimes they even don't bother to look. They ask like questions that are not even, you know, kind in a way. They're just, you know, but still it's worth it because, you know, again, if there's this one person who read the paper and got suspicious, then looked into the data and said, okay, this is kosher, then that's worth it. And if this is a fellow scientist, good, maybe he will give you, once he is asked to review your paper and then he knows, okay, this guy I can really trust, and still, of course, he will look thoroughly, but he won't get the wrong impression in the first place. Or if it's, you know, a lay person, then you convince another one that, you know, the scientific endeavor is worth it. But again, for this, science needs to be open. Again, we get the money from the society, so we need to provide all the knowledge that we are gathering back to them. And also, not only the findings, but I think what we still need to improve is to communicate uncertainties, you know, and negative findings. Because what's in the paper in the end is just what worked, but that's the minority, you know. Usually, you know, experiments don't work. So, but, you know, people can learn from failure and mistakes as well. If they knew, okay, I already tried like a million things and they don't work, they don't have to start over again and sort of waste taxpayers' money. And this is not easy because, you know, I published, for instance, what I did in my bachelor's thesis. I worked on swarming behavior of E. coli, and I published this in the Journal of Unsolved Questions. This is sort of a journal that's also peer-reviewed, where you sort of publish things that, not that don't work, but like negative findings. I found that one component of the E. coli sensory system, nutrient censoring system, and swarming apparatus, molecular swarming apparatus, one component there is not needed for the system to be adaptive, which is a negative finding. But, you know, maybe someone else has the same question and then will also, you know, waste or spend half a year just finding that there is no effect of this component. So I think that's important. And then also there's the second step, you know, now we are in this global viral pandemic. And of course, you know, there's models to predict how many people get infected and what will happen here and there and in the infection in the body. But we only report like mean values, and we don't say, you know, this is the confidence bound. And maybe, you know, it might also well be that nothing happens, or it might be, you know, super dramatic. And, you know, we don't want to make you afraid. We just want you to know there is a chance. It's, you know, it's not very probable, but we cannot rule it out. You know, given all the information we have and all the assumptions we are making, we just can't rule it out. And I think that knowing is always better than not knowing, even if the truth, or what we believe the truth is, or our approximation of the truth, is scary, because it is scary. But it won't change, no matter, you know, how we sugarcoat it. And I think we shouldn't sugarcoat it. We should just, you know, give them the piece of information for them to chunk it and then see whether it's sour or sweet, or I don't know. So I think this is essential. This is crucially important, and I don't have a solution at my fingertips. I don't know, whether it needs another journal to report negative findings, whether we need a different culture of peer review, of publications, of, how people are chosen to become junior group leaders, professors, scientific mentors, science journalists, whether they have to be more involved. I don't know. Maybe probably it's a combination of all of them, or maybe there's even some solution that I don't think of at the moment that might help. But I'm pretty sure. Personally, I think that we need to address this better. Make science accessible and communicate also uncertainties. I think these are the two major points.
Okay, so you already answered another question I wanted to ask. So in our experience, scientists often do not have a lot of time for science communication because they spend their time with either researching or teaching. Can you think about ways to change these circumstances so a lot more scientists can participate in science communication?
Yes. So first of all, that's true. I mean, it's unfortunately true. Also, when I started, Science Slam during my PhD, I basically did this, during nights. So I was sitting in front of the flow cytometer, was measuring cells, and it took like forever. So I had time to think, about this flow aspect that I mentioned. So this is how I started it. And the problem was in the early days, so now we are talking about like 2013, it wasn't appreciated at all. I mean, systemically in the scientific system. Now, what's actually true is that the German Research Foundation, DFG, under the roof of the Ministry of Science and Education, they have funding programs. So if you already have a grant from the DFG, you can basically request another module, it's called, for science communication. So suddenly you get money for it, okay? And then maybe you don't have to do so much teaching assignments to, make your paycheck, but you can use the money from the science communication to have some money and therefore time to do it. So this might be one way. And then another way is, because you mentioned teaching, I think also teaching is not really appreciated. It comes like as a duty if you become professor, but it's not really appreciated. So people, they do the minimum amount because they need to get more third-party funding, God knows what. So they have to publish well, and compared to this, teaching is not important to them, unfortunately. It's not their fault, right? It's just the system that sets these priorities. So if teaching is more rewarded, or there's teaching positions, there might be research positions and lecturer positions, for instance, as it is common in other countries. For instance, in Mexico or India, I know that there's not this stiff like PhD students, postdocs, and then junior group leaders and professors, but there's also dedicated like lecturer positions. And they just do, it's a bit like this. There is teaching professor positions in Germany, but they are not that common, to what I feel. Anyway, so I think what happens mostly is, which is also not bad. Professors don't have time to do their teaching assignments, so they tell their staff, the postdocs. And then they do something maybe that is close to their heart or close to their project, and then they do a very dedicated lecture, a good lecture, because it's close to what is actually done in the lab, and it also covers some basic aspects maybe, and they spend some time and they want to do it and they like to do it, and then you're a good lecturer because you connect to it and you care about it. That's important. And maybe you can use this very good lecture material, and it won't take too much time to convert this lecture to bachelor students, master students, to a lecture for the public, and then you just do there's these Sunday lecture series at Heidelberg University, where the elder come, and then you just use your lecture, slightly modified lecture, to deliver it and share still the same enthusiasm for your project, for your science, but to a different kind of audience. You just do this very step in the beginning that we mentioned and think, now the audience is a bit different. How do I need to change how I tell my story to also make it accessible to them? And this might be another way. But then, of course, the system needs to change still quite a bit. So in Germany, the legal situation, there's this 12-year rule, which basically means that you're on short-term contracts for like half a year, or 2 years. And once they expired, you sign another contract for another 1 year, or half a year, or 2 years. And then there's this rule: if you were 12 years employed in the German academic system, then you're not allowed to get a temporary contract anymore. Because the intention behind this is good because what they want is that then you get a permanent contract. But what happens, in fact, that they never get another offer anywhere, because there's just not enough money for the permanent positions. So after working 12 years in science, in this very uncertain situation, what will be in 2 years from now? Where will I be? Will I still continue? What if you have family or whatever? After this period, when they still, they were 12 years scientists, so they know a lot, and they are so important for this entire system to run and to work, and they are out of work suddenly, and they lost their job, and then what? They are, in their early 40s or late 30s. Then what? Start a new career as what? They were doing science. This was their life, and this is a disaster. This is a huge disaster. So this legal situation needs to change. Again, I'm not saying the motivation was wrong to implement this law. I'm just saying the way it works in reality is just a catastrophe. That's what I'm saying. And if this changes, you can do the math. Actually, I'm pretty sure there's different business models to do more permanent positions, but maybe, one dedicated for people who like to just give lectures, and then, it can be also like 80% position, and then, they have also more time for their family, but they have an income and the permanent contract, they have all the certainty. And then these other 20% maybe can use to pay more technicians and also give them permanent contracts where it's also like very defined. You can well plan this, you don't need this crazy schedule of scientists who never know, long night experiments and stuff. Technicians, they just do their sort of routine work mostly. I mean, no offense, it's super important as well. But for them, this might be a career option because there's people who are looking for just that. And they are still involved in science. Maybe even they publish, So there's all these different models and all this diversity. And so everyone can pick, their own favorite thing without, not at the cost of throwing out people that you invested because you also trained them for 12 years, right? And then you just, they become like, I don't know, cab drivers or something. I mean, and then we are looking for well-educated people to, for PhD students. But if you kept these other ones in the first place, they would be mentors. They can recruit like new people, bring them, teach them. So, wow, it's when I tell this, it looks like science is a huge mess. It's still like the best field I could possibly imagine to work in, but there's so many things that can still be improved. And I think this certainly is one of it. So this Wissenschaftszeitvertragsgesetz, a very German word, needs to change.
Definitely. Okay, so my final question before Lukas takes over. Mm-hmm. You collected a lot of experience in science communication throughout the years. What did you learn through that? Like, is there something you're doing differently now than maybe a couple of years ago?
Yes. First of all, what I learned, where I sort of had the steepest learning curve, was the following. I went on stage the first couple of times. I enjoyed it. I was always thrilled. I'm a stage person somehow. I don't know. So this was always great. But in the beginning, I knew, who is the audience. I thought, like how to do, like how to address them. And I was making some jokes and stuff. And then one night, I did this, and they were like cheering at me, and every joke was, well received. And I thought, great, you did really great. And just the next time you will do it just exactly the same. You think about, target audience, and you do your jokes, blah, blah, blah. And I went on stage the next day, and I did it just the way before. I mean, the same approach. I didn't do the same talk, right? Because I adapted to the new audience, and it didn't work. And I had no clue why. And now what happens is I go on stage, I take a look into the crowd, and I see, there's I don't know, elder people, their students, the way they dress, whether they're on their phone. Actually, I don't know what it is, but I look at them and I immediately know whether this joke will work, how to address them to make them laugh, when to make a break so they will get, excited what's next. I don't know, and suddenly this is something I learned very quickly to evaluate or judge an audience just by first impression. Which again, I don't really know how it works, but this I really learned. This struck me when I realized this in retrospect. It really struck me, and I thought, okay, wow, this is something I wouldn't even have expected to to take from this. And then of course being on stage is one thing, but also if you do other formats of science communication, what is really an important skill is to conceptualize your ideas. To zoom out a bit, see the bigger picture. And once you conceptualize, you can better describe it to any target audience, not just the lay public, also to your collaboration partners who might not be experts in your field, to funding agencies if you write a grant proposal, right? So I think this is really something that you can only learn by practicing it, for instance, through science communication. And then, remind me, what was the second part of the question?
Is there something you're doing differently now than maybe a couple of years ago?
Yeah. So lately, when I started this podcast now, this was actually the first time when. No, actually, even before that. So when I went to Israel, and then I came back to Germany to do some science labs there, and then being in Israel was part of my narrative. And then when these YouTube videos were uploaded from a platform, where they are usually very well received, suddenly I got a lot of, thumb down. Actually, this was the only thing that, they were still like watched like 1,000 times, but I got many, many dislikes. And I wondered why, and it really, I got really, I don't know, it's close to me, so it matters to me. So and then I was sure, maybe they don't like, the research that I was doing, that adipose tissue stuff, or was it the fact that it's about Israel? I couldn't resolve it, right? I don't see who disliked the video for what reasons, but I learned to not let this too close to me, in a sense, And now with the podcast, also there's people that this example that I mentioned before, they don't have any expertise, I think, but still they have an opinion, a very bold one. And then, they tell you that you know nothing and everything you do is wrong for no reason. It's just wrong. And then you think, what you first do, because, you're a scientist and you take criticism, seriously, you think, maybe this guy has a point. Maybe I'm just wrong. Maybe I'm just dumb and maybe I don't get my own research. Maybe this guy is right, which is, of course, Balog, it's just, so this you just need to learn to accept. It has nothing to do with you. It has nothing to do with, it shouldn't affect your self-confidence. It shouldn't affect your modesty. The other way around, if suddenly, people like you for one or the other reason and say, you are amazing and you are the smartest guy I ever met, that's, of course, also not true, right? So be balanced, try to detach yourself a bit from it, which is, it's a double-edged sword, right? Because you want to be close. It's still your science, and it should matter to you. But on the other hand, the feedback you should, of course, it's a way to improve yourself. But if you let it too close to you, and then it might also, affect you. So there, I'm a bit more cautious in a sense, a bit more. I try to be a bit more balanced. But this must not affect the way you approach it. You should not, because this will always come, you should not be afraid of this and therefore change it in the first place. But you should just learn to, or I'm now learning to handle this kind of feedback in a more pragmatic way. Let's put it like this.
Okay, so thank you very much.
Sure.
Lucas and I also talked about social media before, so that really fits.
Yeah.
So if you still have some time, we would love to talk a bit about science now.
Yeah.
Not that we didn't do that before. but could you tell us, relatively short, what your current research at the Weizmann Institute is about?
Mm-hmm. So, as you already said, I was coming from the German Cancer Research Center, where was really interested in the blood system and how blood is generated, quantitative properties, how fast and how come it's so fast, that blood cells are generated every second we're sitting here. It's like 3 million new red blood cells are generated. I mean, that's insane. And I did a lot of deterministic modeling there and proteomics. And then when I decided to go to Israel, I knew two things for certain. First of all, I want to do, again, some joint interdisciplinary work, because also in Heidelberg, I had sort of two mentors who came from different disciplines. And I want to expand my experience. I want to go from deterministic modeling to more statistical models. I want to go from the blood system maybe to the more to the entire immune system. Then also this microbiome aspect. So now I'm working in two labs of Professor Ido Amit, who runs a lab for single-cell genomics. So we do a lot of single-cell RNA sequencing. So then I introduced also the single-cell world into my own universe, and this transcriptomic description of a cellular entity. And the other lab I joined is Professor Eran Elinaf, who is a PhD MD, so he's also a gastroenterologist. And in this lab, we're interested in host microbiome interactions. And there we do metagenomic sequencing. So we sequence all the DNA, for instance, in stool samples where you get genomic content of many, many different bacterial species. Okay, so suddenly you have a metagenome there, which is many, many genomes assembled together. And this we do on bulk, so of course you need many, many individual cells to obviously to have multiple genomes. So this added yet another perspective. And now sort of I have all these tools, experimentally and computationally to sort of address any, not any, but many super interesting complex systems inside our bodies, the immune system and the microbiome. So you also have a lot of metabolites involved in all these things. And then, as you can see, I get easily excited about, science. So then for me, it doesn't really matter the disease context even, or the physiological context we are looking at. I just want to have, some system, a regulatory system, where we don't really understand what's going on. So now also, we are living in a time where data is not actually the limiting step anymore. We can generate data. As I said, thousands of single cells you can, process and measure on omics scale, like the entire transcriptome, or in a very short time, right? So you have all this data, and now you really need to come up with computational models to process this data and to learn some, to see pattern in there, because you cannot just look at it and say, there's this gene, this drives this phenotype. Doesn't work anymore. And also you need sort of experimental models and technologies to sort of mechanistically also validate what you then find with all these computational routines. And this is fantastic because you can basically start generating hypotheses with data that is already there. You don't need to spend an entire PhD to understand a single gene, and only then you can start forming hypotheses what this gene is doing. But you see it more in a more global context. But even the bottom-up approach, so even the other perspective, even if you don't start with a big disease, you can also just start with a general description of whatever system you have. So you get a sample of blood from, you name it, a cohort of your favorite disease, and you just start to sequence it on the single cell level. You see certain heterogeneity, and you find maybe a molecule, and then you have your molecule again, but you already know so much about it that again, this is the birth of a new scientific endeavor that usually you just had, 30 years ago at the end of a PhD or project, and now basically all the projects are starting with this. So it's like super exciting. And you can involve so many people there. You can involve all the MDs who know like the big picture of the disease. You can involve all these biochemists who understand like the individual molecules, how they sort of interact and structure function relationship. And you can involve, you know, people like me who don't know a lot at all, like not a lot of, you know, anything in particular, but a bit of everything. So they sit like in between and listen to both sides like very excitedly, and also the mathematicians who come like with their crazy ideas of distributions and how they propagate throughout biological system. And this is super exciting. So this is the decision I made when I came to Israel, and I don't regret it. I don't regret it a single day that I made this decision, because, you know, there were also people telling me you can't switch fields and you should become an expert in your field. Like you need to find your niche, you know, very precise. But I think that's wrong. I think that's wrong because if you're an expert just in this one protein or one technology, of course you will get the prize for being the best person with this technology or whatever. But then you sort of lose contact to all the other disciplines, and you cannot communicate your findings. And once, you know, knowledge is isolated, it will get lost. So this, you know, relates somehow to science communication. But now I'm talking about science communication inside science, which is also crucial. So, and also I can tell you that from these, so currently I'm interviewing to start my own lab. And from all I can tell, because they are also, right, you apply for a certain position and say, you know, you want to go to a cancer center. But then you come there and say, you know, I did cancer, but I also did like 10 million other things. But there's people who did cancer their entire lives. So why should they choose me instead of them who have like this cancer narrative? And then I'm telling them, you know, but I have all these tools, and no matter what kind of question you will find, you know, cancer, maybe there's some metabolic process, but I know how to handle metabolites. And this is sort of a trade-off, you know, if you want to be like visionary, if you want to develop a corporate vision and also for your own group, that, you know, my group won't be the proteomics group of cancer or something, but my group will be, you know, and also I'm not a method developing guy per definition, you know, I'm very pragmatic. If I need a new method, so in one project now, we had a question that we couldn't address with any technology that was available at the time, so I sat down for half a year and established something that is now suitable to address this question. So I do this if it's needed, but I don't do it, you know, because I can or I want to show off. No, I'm very pragmatic when it comes to this. And what can I mean, interviews are still going on, but my feeling is that people appreciate this. If you sell it well, then usually this sticks to people. Also, I can tell you that they were suspicious. So they say, you know, that's a lot of big words like systems biology, mathematical modeling. What does it even mean? But then once you explain to them, you know, we have these different approaches for different data sets, for different questions. This is something, you know, that helps to convince scientists, because in the end, you know, we're all curious. We all want to answer questions. And if you can help to answer questions, it doesn't matter whether you're not a cancer proteomics guy in the first place. So, yeah.
Okay, thank you a lot. As an iGEM team, we're also interested in modeling our RNA regulatory networks. Mm-hmm. And I think we've been talking beforehand about your experience with modeling in the iGEM team 2013. Mm-hmm. And we wonder if you could be able to share some experiences or even show us what you did model there.
Yeah, sure. So you want me to share my screen? I have the paper open.
That would be super awesome.
Right. You see it? I don't. Okay. Now?
Now, yes.
Okay. So this is the paper that we published back in 2014 in molecular biosystems. And maybe I should quickly explain, because it serves as a good example, this modeling framework that we developed independent of this iGEM team, and as it was not developed by the iGEM team, it was sort of developed, I co-developed it with, it was mainly developed by computational biologists in Freiburg, Breisgau in Germany. And I collaborated with them, so I also contributed a bit to the development of this framework. And it's basically for deterministic modeling with coupled ordinary differential equations. So you basically describe how a certain biological system or components, variables of a biological system change over time, depending on a certain set of parameters. And from these, once you sort of learn your model to the data, or we say calibrate the model with experimental data, you can estimate these parameters, and then from these parameters learn something about the biological system that you model. And in this paper, we were interested in non-ribosomal peptide synthesis. So basically bacteria that are producing small chains of peptides from amino acids. Okay, in a nutshell, that's what it does. But this peptide production is independent of the ribosome, but it happens with a separate molecular machinery. And so, of course, there's a certain mind model, like how this works, which is not important right now. And we also characterize this, like all these different machineries, in like based on their sequences, and then characterize them in terms of growth, which is not important. What is important is how we then use the model, because this was, again, a very pragmatic decision that we made at the time. We said, okay, we have this modeling framework where we can understand systems dynamics. And actually now we want to, since we can measure how these peptides are produced over time and how these bacteria are growing over time, we want to understand what is the property of these kinetics, so of these dynamic changes. And for this we have a framework, so why not, model it there? So let me walk you through. Can you see my cursor?
Yes.
Okay. So what we have in the model here, in the center of it in yellow, is the bacterium. Okay. And this bacterium is coming from nothing, right? This is the symbol for nothing. So it is growing from nothing, okay? And it's growing with these two parameters, beta, which is the growth rate, and max, which is like the maximum growth capacity. So in essence, now you write a differential equation that describes the change of the concentration of bacteria with the change of time, depending on the current amount of bacteria, and then also depending on these two parameters. Now, what do these bacteria do once they are growing? Well, they produce these peptide chains, right? And these peptide chains, they produce from glutamine, right, sorry, I should have known. Glutamine, which first has to become circular. And this process, the more this small circular arrowhead means catalysis. So, of course, the more bacteria you have, the more glutamate they can sort of make circular. So they catalyze this process, which is, of course, done by this molecular machinery of this peptide synthesis with a certain synthesis rate. Okay, K means just, it's a rate constant. Yeah. And in this case, of the synthesis process. Then once you have this circular glutamate, two of these ones have to dimerize. So you also have a dimerization rate. And then basically the peptide we were interested in is this indigoidine, and it's blue. That's why you saw in these bacterial cultures here, they get blue once they produce it. And this was advantageous for us as well, because we could not only measure the bacterial growth over time by measuring the optical density of the culture, but also we could measure the blue color of this produced peptide. Yeah. And of course, everything that has to be produced also has to die, right? So indigoidine is also degraded with a certain constant, rate constant, and then it's also turning into nothing, right? And then we were trying with this model. Of course, we tested different models. For instance, you can now ask, okay, why are bacteria not dying? And the simple question, the simple answer to that question is, well, in the time frame we don't see death, we only see the stationary phase, and sort of death is sort of cancelled out, or you can just explain it by one growth parameter as well. So sort of this was the model that we finally came up with that can explain the experimental data. What is the experimental data? It's the solid line here. No, sorry, it's the dashed line here. So we were basically having these bacterial cultures in a multi-well dish in an incubator, and we're constantly measuring the OD and the indigoidine color, the blue color. Yeah. And then we tested different genetic versions of this synthesis machinery. So this is the wild type, where the bacteria grow to a certain extent. And then, okay, here it seems like also they're going down a bit. And then here you see indigoidine was produced to an intermediate extent and then going down. But then when we tested this other version of the synthetase, so this enzyme molecular machinery that produces indigoidine, bacteria were growing a bit more, but indigoidine was produced even more. On the other hand, when we used this sort of genetic variant T5, there was no indigoidine produced at all. And the model output, so now what the model does, it's basically an optimization procedure where you try to choose the parameters of the model in a sense that the difference between your model simulation and the experimental data is as little, as small as possible, right? Because you want to describe the experimental data. And for this, now we're also coming back to something that I mentioned earlier, we not only have sort of a simulation of the mean, but also we have some confidence bound for this simulation. So this is the shaded gray area. So you see that, you know, sometimes we are off a bit, and actually the experimental data is still like significantly different from what we simulate. But sort of overall, the residuals in terms of the chi-square test statistics, you know, indicates you, given the amount of data points, that we have a proper description of our experimental data, which means that the difference between model simulation and experimental data is reasonably small. Okay, let's put it like this. We don't see qualitatively any difference. It's not the case that, for instance, we don't see any production of indigoidine for T1, which is not what we simulate or measure. So it's always, you know, there's an agreement overall. So we can describe the experimental data. And now we can go and ask questions about these parameters, right? Because this is why we modeled in the first place. We wanted to better understand the system. So we wanted to ask which of these parameters is affected by these different genetic variants of the synthesis machinery. And then we saw, actually, no matter which kind of genetic variant you use, the maximum growth capacity of bacteria is not really affected. It's always more or less the same. I mean, in log space, but okay. But what's super variable is actually this dimerization rate, right? Okay. So it seems like that more the dimerization rate and the synthesis rate, and not the degradation rate, for instance, they are affected by this change in sequence information between these different variants here. So then we sort of more mechanistically understood, okay, what do these sequence variants actually do? Well, they seem to affect the dimerization rate. And then we sort of, to further understand it or even make some sort of inference or prediction, we just simulated sort of the yield of indigoidine depending on different synthesis rates and dimerization rates, because this is something you could then biotechnologically or by genetic engineering you could tune, right? You could generate new synthesis variants, and you ask the question, okay, if with these new variants, if I can really tune this dimerization rate, can I improve the yield, right? And then we basically simulated again the confidence bound of where do these different known genetic variants, where are they positioned in this parameter space? And you see that, you know, T3 and T1 both sort of produce indigoidine to a high, actually T1 is even higher, right? The blue is darker. But actually they are quite similar in terms of their dimerization rate, right? They are very high in terms of dimerization, but what's difference between them is the synthesis rate. So you could make T1 even more efficient if you sort of genetically engineer it to make the synthesis even faster, and then you could even yield more indigoidine. So, and compared to the wild type is a completely different regime. So here the synthesis rate is completely different, and also the dimerization rate. So this is sort of in a nutshell, basically what the modeling helped us here. Again, it was not the main scope of the paper, but sort of it, because then also we experimentally validated more of these different variants. But it helped us again to get at least some understanding of how this system really mechanistically works. So again, in this case, we had a good objective or a good rationale, a good reason to use this modeling approach, and it helped us to understand a bit more about the biological system of interest.
Thank you a lot for this insight. I think it really helps us to get a better understanding of synthetic biology, of systems biology, and how to use it in our project. What I wonder about, we've been looking into different software to do it best, for example, MATLAB or Data to Dynamics, where I think you also worked on. Could you suggest something where we could get started? Can you think about any pitfalls with we could avoid doing because we don't have a lot of experience with systems biology?
Yes, don't overthink it. Just start. It doesn't matter where you start. It just matters that you start. So the reason why you've chosen MATLAB to develop this data to dynamics platform was just at the time there were no good solvers for these differential equations in any other language. But by now, there are even good libraries and packages with solvers for ordinary differential equations in R, which is more like for statistical computing used to be. So now we even have sort of a clone of this data to dynamics in under R. And again, I think it really doesn't matter whether you start with MATLAB or R or Python. They can all be used, and they are in fact used. And so me personally, what I also advise my students to do is not so much, of course, I mean, some people are more like they need a tutorial and a very general introduction into this. And maybe, if you don't have any experience in programming, that's good. But I would start as close to the actual problem that you want to solve as possible. So of course you won't solve your issue in the first lesson, but if you have a problem that is, where you need to look at different sequence variants, then just introduce yourself to the concept of variables and data formats with your actual data. Just start with this, not some dummy data. Already do it with your data, and you will learn so much more on the way. You will get all the basics and all the standards, but you will also start to understand your data. I think this matters more, and just be prepared that, don't be too hard on yourself. It takes time. It does take time, but the payoff in the long run is what makes the difference. So don't worry so much about like, okay, now we even in our lab we have one guy who just works with Perl, and none of the other computational guys knows any Perl, but he does well. I mean, so there is no right or wrong here. It's just, if whatever floats your boat, whatever helps you to answer your question is the right approach. So again, my just pragmatic approach would really be start as close to your actual problem as possible. But maybe, if you're a person who needs more like a general introduction, then it's also no problem. It's really, there's nothing to be ashamed about, or, it's not too late for you to start to learn how to code. It's not that, also doing some stuff in Excel in between, it's not a sin, I'm not saying it's useful or the most efficient way to do it, but it's not wrong. If it helps you to understand your data better, fine. Don't feel bad about this. That's just that would be not stupid actually. So don't just start. If you ask for my personal opinion, and if you want to know, then probably I would go for R, because I find that R, it's well documented. But then you can say the same thing about Python. I would probably not go about MATLAB because it's not free. And of course, at German universities, I think you have campus license, but maybe this is something that speaks about against MATLAB. But again, I don't have a strong opinion on this. So I have a strong opinion about, getting started as soon as you can, and don't be too hard on yourself. But started where and starting how, I think that's not critical in the end.
Okay, thank you a lot. I think that helped us a lot to understand how to get started with systems biology, but also with our human practices, especially when talking about the Science Slam. And I think this interview was really valuable for us. Thank you a lot for your time.
Yeah, sure.
Thank you very much.
Thank you, in fact. And good luck with the project and everything.
Thank you.
Thank you.
Bye.
Bye.
Bye.